Good afternoon, everyone. On behalf of agricultureinformation.com, I'd like to invite the speaker, Dr. Pravallika Sree Rayanoothala, who will speak about exploring fungi's role in creating nanoparticles for practical uses in agriculture. I now request the speaker to begin. Thank you.
Thank you. Good afternoon, everyone. Am I audible clearly?
Yes, you are audible clearly.
Yes, thank you. Thank you. Today, I'm here to deliver a talk on how to explore fungi in the field of nanotechnology. So in the coming 20 minutes or 30 minutes, you will be exploring the strategies, insights, and practical tips related to the topic of the talk. And I hope you will be taking renowned takeaways at the end. Now, let's move on. And the topic is exploring fungi's role in creating nanoparticles for practical uses in agriculture. Me having the background of mycology, plant pathology, my insights are little inclined towards plant disease perspective in broad sense, not all the agriculture thing, but mostly towards plant disease perspective. My slides are not moving. 1 second.
You can stop sharing the screen and restart it.
Yeah. Sorry for interruption. And in this presentation, this first slide gives you a overall index kind of thing. You'll be learning about how nanoparticles, nanotechnology, and fungi-mediated nanoparticles application and their mechanisms of biosynthesis and applications in crop stress management and their perspectives and challenges as well as future outlook and concluding remarks of fungi mediating nanoparticles will be addressed here. From the right picture, it can be understood that nanopesticides, whenever it is used, the benefits of using nanopesticides are target-specific, coated or primed with silver nanoparticles, which have increased shelf life and, more importantly, its stability. Next, in this slide, we'll be learning about various applications of nanotechnology in agriculture. Nanotechnology is undoubtedly playing an important role in reviving the agriculture and food production industries these days. Nanoparticles have proved to be a promising alternative to the manufacture of nanofertilizers compared to conventional fertilizers. Here you can see near these nanofertilizers and nanomaterials, which can reduce excess chemical fertilizer usage, thereby controlling environmental pollution. In here, the various applications of nanotechnology in agriculture development were described in various fields like crop improvement, crop protection, even in precision farming as well, and stress tolerance using nanoparticles under various stress like drought, salinity, and UVB stress. And everything is shown here in a descriptive, in a tutorial manner. Next, moving on, and this is the application of nanoparticles in agriculture. Previously, it was shown like nanotechnology, its wide, broader sense. Here, this is nanoparticles. Here, it is illustrated that nanoparticles can be used as protectants, carriers, after which the advantages in relation to pest and disease protection from use of nanopesticides, which I have just mentioned in the first slide itself, where the benefits like increased shelf life, specific uptake, like it will be like site targets specific, and it will be having higher solubility and decreased soil leaching. This is a problem facing by farmers these days, and toxicity also will be low, which mentioned earlier. And why we need to shift towards this nano usage, nanoparticles usage in agriculture, maybe in the form of under the field of crop improvement or crop protection. With the hint of this all background, whatever I had given, like application of nanoparticles, nanotechnology, which describe the advantages of nanotechnology, I hope now it will be going smoothly right now into the topic. Now, undoubtedly, all the plants are encountered by many fungal diseases, which are about 70% of all major crop diseases, and as well as the annual crop losses due to pre and post harvest fungal diseases exceed even 200 billions now in India itself. So, at the same time, the spent or money spent or economically, if you see, like spent on fungicides also, it is increasing day by day. It's directly correlated to environmental pollution as well. And this impact of nanoparticles on crop plants is rising area of research with all these concerns that needs to be meticulously explored. And in recent years, these nanoparticles have been engineered particular attention as a potential candidate. However, these applications are still in its infant stage only. So this should be like always taken as priority for unforeseen health hazards and environmental concerns. And it is understood that the use of pesticides in agriculture is becoming more hazardous day by day, as mentioned earlier. In order to replace such toxic materials by equally useful substances is definitely an excellent choice. In place of chemical fertilizers, you can go for nano fertilizers and so on. So it is also especially easily available silver nanoparticles, which are antimicrobial for most of the fungal and bacterial diseases in plants as well as for humankind also. And it is useful if a better protocol for its application is developed. Now, let's get into the core topic, fungi mediator nanoparticles. Here, these are few, I can say, heroes in the fungal community, which are extensively used as nanoparticles, like Aspergillus species. And here you can see Coriolis is a mushroom, Fusarium, yeast, trichoderma, like yeast like Saccharomyces and Hansenula, Candida species, and Verticillium species. And the next slide, you will be seeing these are the examples of recent metals isolated from various fungi like platinum, iron, silver, copper, gold, and on. Here you can see the fungus which we selected for this isolation of nanoparticles and their application as well as their differences. So moving on. This is most important. First of all, we have to know what factors influencing the nanoparticle production. So here, I just concentrating about the fungal mediator nanoparticles, and hence we are just differentiating into silver nanoparticles and gold nanoparticles. Here you can see, of course, like major work has been done with silver nanoparticles than gold nanoparticles produced by fungi, extracellularly or intracellularly. And of course, the factors like the particle size always is fungal specific and also metal specific. That means like which you are using salts to isolate and all. For example, the silver nanoparticles produced from the interaction of Aspergillus may not have the same dimension as those produced by Fusarium. Even if other conditions like concentration, pH, temperature are identical, and also the most important is incubation time. Usually it will be from 15 minutes to 60 minutes, but exceptionally trichoderma species, it takes about 72 hours, but it is very useful for large scale production of nanoparticles. And their size also, it will be this normal, like 5 to 50 nanometers. And whereas for gold nanoparticles, apart from these factors like fungal species, temperature, pH, metal, and incubation time, color is also a factor for assessment. Like, so we have like different colors showing different sizes. For example, this biosynthesis of gold nanoparticles from fungi, of course, like it's a review, it's like reviewed recently only. It's not like old as silver nanoparticles research. They are resistant to oxidation and dispersed very nicely. And the color always corresponds to the particle size, as I mentioned earlier. For instance, like yellow, red, and different colors will be there. Like yellow will be giving you the large size of nanoparticles, whereas red, like small, and some are like dark pinkish kind of thing will be giving you the fine nanoparticles. of varying size and morphology impact. So here the right side, the reaction you can see, if suppose uh, when this gold chloride, trichloride is dissolved in water, the reactions will be like occurring on mycelia of fungi, which contains proteins and all. So it requires 3 electrons and reduction occurs in 3 steps, which is depicted here. And this, the fungi mediator biosynthesis of nanoparticles. Like how will the fungi produces or biosynthesize the nanoparticles? That can be extracellular or intracellular or membrane bound by yeast. And generally, this biogenic synthesis of metal nanoparticles involves bioreduction of metal salts to elemental metal, which may be stabilized by organic molecules present in the microbes. That means like fungi here, it is helping to break down from the metal salts, using metal salts to elemental metal. If you can see in the last reaction, here also you can see the silver, it came to metal silver after reduction and all. So likewise, here we can say the other way of producing metal nanoparticles is also like absorption. Like fungus absorbs the metal ions in aqueous medium. Like earlier I mentioned, that reaction would be done in water, aqueous medium, which is bounded to the surface of cell wall of the organisms. Here it's fungi. For large-scale production of nanoparticles, fungi and yeast are preferred over other organisms, usually. And the yeast example I have mentioned earlier, Saccharomyces candida species also. When fungus is exposed to metal salts, like I told silver nitric oxide or gold chloride, so it produces, first of all, the enzymes and metabolites to protect itself from unwanted foreign matters. And in doing so, the metal ions are reduced to metal nanoparticles without affecting the growth of fungi. That's why fungi and yeast are these days the promising organisms from where you can isolate the nanoparticles. Of course, the exact mechanism of intracellular synthesis of gold and silver nanoparticles is not known yet, but it is for sure that in the presence of fungi, they are formed on the surface of the mycelia. That is already found in the research studies. It is proposed that the metal ions in the solution are attracted towards fungal mycelia. Like suppose the positive charge, they'll be having positive charge in them and slightly negative charge on the cell wall, due to which this enzymes, amino groups of protein followed by reduction will happen. So this is the mechanism of biosynthesis of nanoparticles. Intracellular also, usually the gold nanoparticles that can be stabilized by substances, like even we can use from externally also, if they are not producing enzymes, so we can induce this catalase or ascorbic acid as well. So basically, this both intra and extracellular synthesis of nanoparticles by fungi is done in a very simpler manner. And this gold ions are trapped by proteins and enzymes on large surface of the fungi, which I also mentioned just earlier. So they'll form the aggregates of larger dimensions. So moving on, here I just gave some examples of intracellular biosynthesis. Here it's a verticillium species isolated from the Taxus plant. It interacted with the silver nitric oxide at certain temperature for about 72 hours. Here the incubation time is also 72 hours. So the process and all was explained earlier. Like this is the same thing. After interacting with this and positive and negative charge also interacts and produces enzymes and other metabolites, which will aggregates into larger dimensions, and thus it will give us the metal ions, silver ions, for example, like silver ions or gold ions. Next, moving on, and here is a pictorial representation of biogenic synthesis of silver nanoparticles, where we can see here, although like many studies have been published concerning the biogenic synthesis of silver nanoparticles using fungi, But the specific mechanisms, this is like apt mechanism. It's not like have not been fully elucidated. It is known that extracellular synthesis of nanoparticles occurs according to reactions in which enzymes present in the fungal filtrate. So it is basically dependent on fungal species. That's why it's like indirectly I told like fungal species is the most important, because the enzymes are most important to occur or to induce the reactions to produce the nanoparticles. So the fungal filtrate enzymes, and thus they reduce to silver ions, producing elemental silver at nanometric scale. Of course, like as I told, the size depends on the synthesis conditions like fungal species, temperature, pH, and also the medium, as well as the coatings of nanoparticles, like whether that's silver or gold, whatever. So moving on, here we'll see applications of silver nanoparticles, specifically silver nanoparticles, which can be synthesized from fungi. So here you can see many studies of biogenic synthesis of nanoparticles using fungi have shown results that are very promising in agriculture these days for the application of these systems in controlling pathogenic fungi and bacteria. Also, they'll have the bactericidal also, and also combat, even if you take this humankind, it can combat the cancer cells and virus, and it can provide larvicidal and insecticidal activities also. It's like all in one package, silver nanoparticles applications. And here very interesting organism of fungi is macrofungi, that is mushrooms. Mushrooms also produces particles, and so far more than 70 mushroom species have been examined. And here it depends on the same fruiting bodies and the content of bioactive compounds, which plays a major role in production of the silver nanoparticles. Both the edible and as well as the medicinal mushrooms have gained more popularity, and research is going on a very large scale right now than other silver nanoparticles, which are known for their antibacterial properties, which I mentioned in the last slide, which showing a promising application in medicine as well as without any toxicity in both human as well as the plant life. So concluding remarks of this most innovative and new research is biological synthesis of nanoparticles is completely safe and relatively inexpensive. The synthesis of metallic nanoparticles using microbes had been shown to possess a strong potential with various applications. And this utilization of fungi to fabricate nanoparticles has been greatly considered, which have the several advantages I mentioned, including like easy handling and downstream processing as well. Like you can just isolate, recover, and purify, and you can react with the soils, and therefore you can find the metal nanoparticles, which can be used in agriculture in various fields, crop improvement, crop production, protection, everything you can use. And however, the interest of several researchers had been towards the micro nano fabrications of nanoparticles, like they are more interested in exploring mushrooms in this field. Of course, this micro nano fabrications of nanoparticles has been considered to be very satisfactory method for synthesis of various types of nanoparticles, not only silver, gold, and all. Which has equally lot of potential for disease reduction, or else in full agriculture stream, and also food industry, style industry, medicinal industry, cosmetics, and various optical and electronics and also. But in the same way, we have few challenges and way forward in this fungal nanotechnology. That is, the major problem facing fungal nanotechnology is under exploration of fungi. That means like research should be more on this to explore fungi. As stated earlier, only few fungi are well documented for their nanoparticle production, their mechanism, potential, and also their biology at large scale. And this has somehow limited the full exploration of great kingdom of fungi. Some fungi are not even culturable in laboratory because the incubation conditions we may not provide, or else the media requirements are not fully understood. Or else the whatever the media we are producing, that may not be met the requirements of the fungi to grow. Furthermore, the optimization of fungal growth and nanoparticle production has always remained a major challenge in the option of microbial nanotechnology. So I hope like we should do more research on fungi exploring in this area. Generally, this nanobiotechnology faces the challenges of public acceptance, that is for sure, limited research on long-term exposure to nanoparticles, and little this legislation guiding also nanoparticle application in most countries, in most developing countries as well. So this microbial nanotechnology will definitely solve these problems, but the problems are still abound because of these above mentioned things. So besides this, of course, this titanium dioxide, which is well known for this nanoparticles, and also iron oxide, which were that already applied in this food industry, this coloration and all. So no other nanomaterials are included in human food except this titanium dioxide and iron oxides. So for this, we should use for that education of humans on the safety and benefits of nanoparticles is very essential. And the future outlook is mentioned here. The nanoparticles, whichever derived from fungi, have great potential for a wide range of industrial and pharmacological applications as well. It's like broad usage. As I told you, it's like all-in-one package kind of. And though a range of fungal species have been tested to synthesize nanoparticles, there are many edible, like mushrooms and all, which can be explored. Because fungi, why we should concentrate on fungi? Because it themselves, they have very essential role in this ecosystem, which are well known for its decomposition and also nutrient cycling. Therefore, the mechanisms used by fungi or mushroom for natural process could be exploited in the near future for sustainability, which is very essential these days for future generation. And thank you. Hello.
Yeah, thank you, Doctor. So I'll now take up some questions. Just 1 second.
Yes.
So the first question is, how can fungi be harnessed to produce nanoparticles that benefit agriculture?
Yeah, this I have mentioned here already, the mechanisms of isolation and all here. You should just interact with the salts. Like here, I told this silver nitric oxide. So thereby it will undergo such downstream of process, reduction and all. At last, it will be giving you in which the silver coated nanoparticles. I'm just telling you this usage of the silver nanoparticles in plant disease. Suppose the plant is infected with some fusarium wilt or verticillium wilt, though the pathogen can be isolated for nanoparticles. So first of all, this concentrates on the fungal cell wall. So it breaks down the cell wall. So therefore, many enzymatic reactions and all biochemical will be hampered. So therefore, the infection, the growth of the fungus and everything we can halt. So this will be like greater use if we use in this plant disease, and very economical as well as environmental friendly as well. Hello.
Yeah, okay. What specific types of nanoparticles can fungi generate for agricultural applications?
Yeah, usually the broad sense we can say silver and nano, silver and gold. other things, iron oxide, platinum, but still in agriculture, we'll be using silver and gold mostly. But in other industries, we'll be using titanium dioxide, iron oxide, and different things. Yeah, here the applications also mentioned.
Okay. Are there any challenges in scaling up the production of fungal derived nanoparticles for farmers?
Yeah, for farmers, it's a challenge only. But for researchers also, there are few challenges. Like I have mentioned you, few fungi are not culturable, and that should be like highly advanced labs, which we can provide the incubation period, and we can try the new media
We have lost your voice. Hello, Dr. Pravallika.
Hello.
Yeah, we lost your voice.
Okay, sorry. So from where? Okay, that's fine then. So definitely the challenge for farmers. But if the research is going on a large scale, and if we can produce more number of nanoparticles, the extension department, we have various KVKs and all, or even from direct research labs. If you take this mushroom labs, will be like various places, so they can directly connect to farmers. But still, that will be like we need to educate or create some awareness because of this. Now the seed treatment, chemical seed treatments are already like, it's just very easy for farmers to follow. But this nano priming, this thing will be taking some time, but still we can achieve this after doing a little research on this. Yeah. Hello.
Okay. So next question. How do fungal nanoparticles compare to traditional agricultural inputs in terms of cost effectiveness?
Cost effectiveness, basically normally the cost management thing will be production of nanoparticles that can be using any method. But when you use a fungi, the extra cost is your lab, your fungal cultures, media and all, but that is not that much significant to be considered in terms of cost effective. So this is like extra cost adding thing. It's just nominal thing.
Okay. And can these nanoparticles help improve soil health and nutrient availability for crops?
So if you are using nanoparticles for soil improvement, then it is definitely a target specific. No doubt it is not like environmentally harm. Here, one second. In my first slides, so here you can see, whenever you are using this nanoparticles in whatever as a protectant or carriers, whatever, it will improve the efficacy of that particular chemical or whatever. But the thing is, the soil leaching, if you are using the normal seed treatment of fungicide, that can cause or harm other beneficial microbes in the soil system or in plant ecosystem. But this nano fertilizers or nano pesticides will not do that. So in that way, this will definitely increase the efficacy of whatever you are using, protectants or fungicides or whatever. And toxicity will be also less, but efficacy will increase definitely. And it will be nutrients also, it will be bioavailability always.
Okay. And are there any potential risk or environmental concerns associated with using fungal nanoparticles in agriculture?
Environmental will not be there because fungi itself is biodecomposers and all. So definitely it will not be leaving any residues in the environmental, either in the soil or in environment, atmosphere, nothing. It won't be there.
Okay. And what crops or farming practices could benefit the most from the application of fungal derived nanoparticles?
Yeah, mostly right now the cotton research is more in this. Cotton diseases are mostly their experimenting level. They are doing mostly this lint crops, oil seed crops and all. They are experimenting right now. But I don't think so that will be a challenge or any constraint in crops usage. I think we can use for any crop of this.
Okay. And how do fungal nanoparticles impact pest and disease management in agriculture?
Sorry, please pardon.
Yeah, one second.
Yeah.
How do fungal nanoparticles impact pest and disease management in agriculture?
Yeah, firstly, I just mentioned with an example as well. This fusarium, if you take, you will just constant the mode of action of this nano fungicide will be always the cell wall of the fungi, which breaks all the biochemical steps in of fungi, which will hamper its growth, like its mycelial growth can be there. It can't produce the aprosorium as well. So hence it will not grow, and in the population as well, severity, virulence, everything will be breakdown because the cell wall will be breaking down. The mode of action, the target thing is the cell wall. So various enzymes and all will be breaking down. So the disease can be controlled with this. And it will have a good shelf life also, as we use elicitors in the same way, in the same mechanism this use. But it will not be the systemic, but still we can use as nano fungicide, which will concentrate on breaking down the cell wall of fungi.
Okay.
Yeah.
And are there any successful case studies or real world examples of farmers benefiting from fungal nanoparticles?
The farmers level might not be there, but in this table, you can see all these are success only. The fusarium, usually the fusarium is mostly explored in this field. Fusarium oxysporum, they'll be isolating not only here, I mentioned platinum, but there are many silver, gold nanoparticles which are isolated from fusarium species, which are using in various fields of study, which are successful only. Farmers level, that is a gap actually. I've mentioned this in the last step. So if that is diluting, I think farmers also will be taking up this thing, nano priming concept.
Okay. So now we have the last question.
Yes.
What resources or support systems are available to help agri-entrepreneurs integrate fungal nanoparticles technology into their farming operations?
Okay, the resources, like you can say every like nanotechnology labs, wherever they're concentrating this research. So definitely, I think suppose if in our university, if you take like even in West Bengal or Hyderabad, they'll be doing in the department of this nanotechnology, and they'll be through extension like KVKs and all, they'll be reaching farmers. For farmers, I'm telling you, so they can just provide some samples so that they'll be like mini kits kind of concept, like samples they'll be providing, and they'll be doing field trials and all. So like research labs, you can just contact the entrepreneurs and all.
Okay. Okay, Dr. Pravallika, I think now we have come to end of question round, because I don't find any more questions from participants. So on behalf of agricultureinformation.com, we'd like to thank you for the detailed presentation and answering all the questions in depth. And we also like to thank all the participants for joining this meeting. The meeting will now be closed.
Okay, thank you.
You're most welcome. Thank you.